In this video, I'm going to talk a little bit about the viable cell count. So viable means that it's alive. And what we're doing in a viable cell count is we're trying to count the number of living cells in a culture. So what we're going to do to count the number of living cells is we serial dilute them, and then we're going to plate the serial dilutions and count how many cells grew. And the way that we count how many cells grew is by counting how many colonies we see. A cell that's dead can't grow, obviously. So we can count or differentiate the dead from the living cells in a culture just by counting the number of colonies that grew. So when you look at these plates here, you can see the dilution has occurred and we've already grown these up. So here's a 10 to the negative 5, 10 to the negative 6, negative 7, and negative 8. And you do see some contamination in the background, but we want to focus on the larger white colonies. And we want to identify a plate that has between 30 to 300 colonies. If it's less than 30, the math can't be accurate, and if it's more than 300, typically the colonies are too close together to be able to differentiate them. So again, it'll be inaccurate. This one here looks like it has far too many colonies, so we would likely choose this plate to count. And then an easy way to count these would be by using a marker and just tapping on each one of the colonies as you count it and keeping track from there. So we're not going to actually count these plates, but I am going to talk about the math behind this. So when we do serial dilution and we want to calculate the original concentration of living cells in that original culture tube, we need to take into account the dilution factor. So let's say we ended up with 67 colonies, and this was on the 10 to the negative 6 plate. So we had a 10 to the negative 6 dilution factor. I'm going to make this a little bit bigger so you can see it a little bit better. So in order to solve this, we would do 67 divided by 10 to the negative 6 in order to get our original cell density. Now you could try to do this in your calculator, but there's an easier way to do this. You would write down the number 67 and then put the number of zeros down as the exponent here. So we have an exponent of 6, so we're going to put 6 zeros down. And this is our original cell density. So this is the final answer. Now it also depends on how much we plate. So typically we're not going to plate a full milliliter because a full milliliter is going to just flood the plate. So we typically plate a fraction of a millimeter. Now what I have here written on the plate is the final dilution factor. So 10 to the negative 6 is the final dilution factor for this plate. But let's say we plated one mil of a 10 to the negative 5 dilution, and I'll abbreviate that DF for dilution factor, and we counted 100 colonies. Because we plated an entire mil, this is our final dilution factor, and we're going to do the math like we did before. 100 divided by 10 to the negative 5, and that's going to give us 100 with 5 more zeros. And that would be our final answer, 10 million. But if we plated 0.1 mils, which is a more likely scenario, of a 10 to the negative 5 dilution factor, and we get 100 colonies, what we would do here is, because this is a fraction of a mil, we actually have to increase the dilution factor by 1 exponent. So our new dilution factor becomes 10 to the negative 6. So now we would write down our 100 for the 100 colonies we counted, and we have to add 6 zeros. Now why did we do that? Again, because we plated a fraction of a mil, we have to account for the fact that we're not plating the full mil anymore. We're only plating a tenth of a mil. When we plate a tenth of a mil, that actually bumps up the exponent, the dilution factor exponent, to the next one up. So in order to solve this one, if we're plating 0.1 mil, we would have to increase the negative 5 to negative 6, and then in the end we're going to be adding 6 zeros to it. The math is going to be identical, so we have 100 divided by, except the dilution factor we're going to use is 10 to the negative 6. Again, you can do this in your calculator just to confirm, but it's going to work out that that's how many zeros you add, and it makes it a lot easier to count the colonies. This scenario up here is your final dilution factor. This is telling you you're plating 1 mil, and this is telling you you're plating 0.1 mils. If you plate the full mil, it is your final dilution factor, just like the first example. If you plate a fraction of a mil, you have to take into account that you've only plated a small amount of the full mil. So a tenth of a mil will increase the dilution factor by 1.